I want to move on and talk about, what's limited by challenges in deployment. So, innovation and invention, they're often limited by that, by the challenges in deployment. And so I want to talk through how the field is evolving around the scale up of production for these novel engineering technologies as they impact both near term and long term deployment specifically. Um, Sam, can we start with you on that?
Yeah, I think when we think about new nucleic acid therapeutics, we've learned a lot from the linear mRNA examples. As some of these novel formats, come to the fore. I think we need to really account appropriate timelines for process developments. That's what we're seeing at Nutcracker, because every time you change some aspect of manufacturing or you change the format, we tend to see some unknowns that become manifest. So, for example, side product formation that you weren't necessarily anticipating, and then that pushes you to think, what's your purification scheme going to be, so that you can hit the kind of quality attributes that will be necessary to deploy this product in the clinic. So things that we have learned from mRNA manufacturing is that their quality profile is very sensitive to a number of parameters during the manufacturing, as well as the consistency in the raw materials that are used. So typically the approach that we follow at Nutcracker is that we drive very hard to use our manufacturing platform that we call our NMU, where everything is, as I said before, is produced on microfluidic devices, and the process is all kind of driven by software, and we use this really to minimize the run-to-run variation. And that's not something that we achieve immediately for a new process, right? So we have to see, okay, where's the variation come from, and after a few runs, we tend to see that variation come down to the point where we can run these processes very stably. And then once you start that as your foundation, then the next steps that we follow is we start playing with each of the variables. So which variable kind of impacts what aspect of the quality attributes, and kind of learn. How can we minimize some of these potential impurities? And we've seen that changes in the process help you achieve that to a large extent. But also you might need to think, okay, no, this is an impurity that is not going to be acceptable in terms of its activity or safety profile, and we need to then think about, okay, how are we going to get rid of it, and how are we going to get rid of it in the context of our microfluidic devices, which adds an extra level of complexity. But we've been through this multiple times quite successfully, and I think that with new formats we'll be able to achieve this too, but it's not something that will happen immediately. So we need to think about giving ourselves sufficient time for the process development step, and to do that in a very systematic way. The aspect of this that again people tend to minimize, but that we found to be very important is on the design up front. We've seen that some sequence attributes are actually quite predictive of the quality of the product. So can we screen from that for those things upstream, and then kind of this concept of quality by design is something that we're very excited about in Nutcracker, and that we've seen it to be very powerful. That especially when we're thinking of proteins that have a little bit of higher complexity. So things, for example, where the protein design requires you to repeat a certain protein domain multiple times. Those things will be more challenging to manufacture. Or the other setting is in the personalized setting, right, where to keep costs down means that we probably won't have the luxury of making turn versions of a personalized product for a patient. You need to be able to use your design technology and driven by machine learning to kind of come up with a design that off the bat is very likely to yield the quality profiles that are necessary for the release of a product in a personalized setting.
Good. Okay. Thank you. And Nathaniel, before we hear your commentary on the exact same topic, I want to ask Miles to please post the poll question for our audience. Audience, please take a few seconds to respond to that, and we'll post your responses soon. But Nathaniel, I wanted to ask you the same question. How does the evolution and scale of production of novel engineering technologies really impact the near-term and long-term deployment?
No, absolutely. So I think what was interesting is just how much evolution in the space we've seen over the past few years. And what's nice about RNA technology is that, if you're creating RNA for RSV versus EBV versus flu, it's fundamentally the same manufacturing process. So that's what's attractive about it. What's deceptive about it is when you start bringing all these new modalities that have been engineered, it becomes a lot more complex. And sometimes people can fall asleep at the wheel because of that simplicity. And I think that Sam was starting to allude to a lot of those things. So, a lot of processes that were originally built for small interfering RNAs or ASOs, just things that are very small, started to be used in those same manufacturing processes for longer things like linear mRNAs, which tend to be somewhere 1,000 bases, 3,000 bases, that range, to now very long RNAs like self-replicating RNAs that can be anywhere from 9,000 to 16,000 bases. And that poses a bigger engineering challenge as you're doing production than I think people appreciate. So the analogy I would use is, imagine you have a dirty box of Legos, and I say you need to clean those Legos after clicking some together. Click 2 together and start cleaning them. Click 20 together and start cleaning them. Click 200 together and start cleaning them. The way you clean them is going to be very different because the ones that are just 2 blocks are much more stable. The ones that are 200 blocks are a lot less stable, and that's just because of the biophysics of what you're dealing with. I mean, it's the same component pieces, the same thing that you're doing, but you're going to have to take a different strategy to doing that. And I agree wholeheartedly with Sam in that you need to take the time for that process development to optimize each of the parameters and the input to it. And, the field is just catching up, in that sense. So one of the first things that we did was to form strategic relationships with different CDMOs. We work with Curia and Lake Pharma, or Precision Nanosystems and Danaher, and forming those relationships early on so that you can really build this end-to-end integration as you're creating new manufacturing processes, I think is pretty critical to the longer term. So shorter term, you can produce some of this material a little bit more easily with these new modalities because there are specialized facilities for it. But if you want to be able to get to commercial deployment within a reasonable time frame, you're going to have to work with some of these larger end-to-end integrated organizations and figure out ways to integrate some of these new technologies and optimize these parameters. And it can be a pretty long road, even if you know the pain points of it. So I think that, in the next 5 to 10 years, I expect there to be a lot more options in terms of CDMOs or other partners that people can use in the space. But right now, I think people are trying to cobble the pieces together and string them together so that you have the entire manufacturing process, and that can start to be pretty complex, especially as bringing in some of the newer technologies, which will be beneficial, like microfluidics that Sam was bringing in, but just may have constraints around whether the CDMOs can use them, whether they have the personnel and the expertise to be able to integrate those. And so it becomes a jigsaw puzzle of manufacturing, and you have to have both a short-term kind of eye on that as well as a longer-term eye on that as you build up these new technologies. And each one has their own pain points. For these longer RNAs, in addition to the biophysics, they can start to have more secondary structure, as they start to be produced. And so it's just things like that. A circular RNA, obviously, it's a new process where you need to figure out how to, tie the 2 ends of the RNA together and the ways in which people do that, and, the individual components to make that actually an efficient process. So, all of these modalities are going to have their individual challenges, even though on the surface, the actual steps themselves look very similar. And so I think that's the real challenge for our field.